Okay, we're gonna finish up kind of here with chapter 16, talk about review transcription control and additional controls in eukaryotes, talk about gene expression with complex traits, and with cancer. I'm also going to review PCR and talk about DNA sequencing. So let's get right into it. PCR, polymerase chain reaction. It's really just one enzyme working in a test tube, and that enzyme is a DNA polymerase from Thermus aquaticus. So it's a heat-stable DNA, and we put that in with the DNA we want copied, and then just the raw materials like primers and nucleotides. And then the thermocycle, which is a glorified hot plate, cycles through 3 temperatures. Okay, and these cycles, one of them is hot enough to get the 2 strands of DNA to separate. Then we cool it down to the primers bind, and then we cool it or we heat it back up to the temperature that the DNA polymerase likes, and we copy DNA. We elongate strands. And so each cycle takes maybe 2 to 3 minutes and doubles the amount of DNA. So you run 30 cycles and you got 1 billion strands of DNA. Very, very nice. Okay, easy to do, not that expensive. Okay, a very similar process can be used for DNA sequencing. Here we use, again, DNA polymerase and nucleotides, but we also include chain terminators that stop DNA polymerase from adding further bases, so it makes a fragment, but that fragment will end with one of 4 fluorescent dyes. We then separate those fragments by size with electrophoresis, and a computer uses a laser to identify each fragment, what it ends with, what base it ends with. So looking at this, at the lower right, here we have 4 different colored chain terminators. The red is for thymine, black is for guanine, green is for adenine, and blue is for cytosine. And then we see peaks of different colors when we separate the fragments by size. And so if you look at just to the left of 120 on the lower right, the first uh, nucleotide at that spot will be guanine because it's black, and then green, which will mean adenine, red, which then means thymine, then 3 different fragments that are green. So adenine, adenine, adenine. And so the machine does this. So you just load it up and then you go watch cat videos. Sequencing can be done for individual genes, which we identify with a probe, or whole genomes. Very good for identifying species, for identifying individual organisms or people. It can be used to diagnose infections. We can find out which genes are active using this, just modifying the technique. So that's a gene array. We can identify mutant genes and like disease risks for like BRCA. We can type tumors. We can look for genetic diseases. This can be used in criminal justice. Powerful technique. The machine is more expensive here because it's got a laser and gel electrophoresis on, but it's reasonable, affordable. Okay, so one final issue to talk about is the safety of these technologies. And most of the concerns about safety have been about people eating food or taking medicines that are genetically engineered. And might that change us? And of course, we've been doing this for 30 years, and millions of people have been consuming foods like this or taking medications that are made like this. And so we think it's pretty safe. The stuff is tested. There's really been no side effects. And really, these altered organisms that we eat, they have one piece of DNA inserted in them, and they produce usually just one different protein. Okay, we digest that just like we digest all DNA and all proteins. These fears are imaginary. And so, here we have kind of imaginary frankenfoods. The dangers are way overblown. To me, a bigger concern are ethical issues. Um, who decides what DNA we can use, or who owns it? Can you patent these things? And the answer is yes. Who benefits? What if we make medications based on DNA testing so expensive that nobody can afford them? Okay, a good example is the BRCA gene. The test for that is owned and it's patented and it's expensive. It's not a generic test. It's a proprietary test. It's more expensive than it should be. If you click the link here, they'll talk about the devious defecator, where an employee is, or an employer is trying to force employees to get tested to identify somebody who's doing something annoying but not really important. So it's kind of an important consideration. Okay, so the last thing I want to talk about is then these regulatory things. And remember that DNA is the same in all cells, same genetic code. The same processes are used to transcribe and translate them, the same enzymes and enzymatic processes. These things are controlled by proteins that bind specific sequences. DNA binding proteins like helix turn helix, zinc finger, leucine zipper, they can identify specific DNA sequences. And so the repressor here in the lac operon for prokaryotes identifies a sequence on the operator. The cap protein, which is a inducer, identifies specific sequences. And these things also interact with other proteins. And so like in prokaryotes, the lac operon is the standard molecule or model for gene control. Normally it's repressed by a repressor protein blocking RNA polymerase. But if lactose is present, the repressor lets the operator go. And if the cell is low on energy, the catabolism activator protein helps RNA polymerase bind to DNA and turn things on. Eukaryotes are a little bit more complex. We have other mechanisms that we don't have in prokaryotes. We can methylate the histone proteins, which causes the DNA to pack tightly. So it essentially just turns those genes off for long periods of time, maybe more than one generation. We have the same transcription factors, but here even more complex, even more of them that can turn genes to repression or induction, very specific. We can also edit RNAs by adding a cap and a tail, splicing them. We can degrade them at different rates. We can even degrade proteins. A good example of regulatory control of eukaryotic DNA is the transcription factors that control development. These are the Hox genes in animals. They control things like the development of body regions, organs, and limbs. This allows multiple genes to work together by all of them sharing transcription factors. In addition, this is really crucial in cancer therapies. Okay, now we know that things like oncogenes and tumor suppressors, these genes, the structural part of these genes can be mutated to cause cancer, but it could also be mutations to the gene expression and epigenetic regulation and RNA stability genes, that these things could change the levels of these molecules and change the abundance of these proteins, maybe to the point where they're not available at all. And so when we talk about cancer genes, we're also talking not just about the structural genes, but the regulatory genes. And this provides us with new targets for cancer therapies. We can look at epigenetic changes that might affect the cancer-causing genes. We can look at changes to the regulatory RNAs, like microRNAs and silencing RNAs and the RISC gene. We can look at overexpressed proteins, and all of these things are done in tumor typing. So it's not just the oncogene or proto-oncogenes or the tumor suppressor genes, it's these regulatory genes. Okay, so that gets me through recombinant DNA and DNA sequencing. I want to talk a little bit more next week about genomics and proteomics, but we're largely done with our chapter 16 and chapter 17 section. And so we'll probably start reviewing on Monday for the final exam.